Hello friends, let's talk about a new topic today, that is a plumbing system of the cell. We know there's a places in the cell through which the water enters, and those places we call it as a protein, and the name of that protein is the aquaporin. And these proteins were discovered by the Peter Agre, who fetched Nobel Prize for it. He was working on RH factor, and he suddenly discovered these proteins, and he called it as these proteins are going to serve as a plumbing system of the cell. Let's talk about this protein in little more detail. These proteins, we call the aquaporins, they're the family of the proteins which are spanning the membrane several times, and the molecular weight of a monomer, which is going to be tetramer, we'll talk about it little later on, around 30 kd, and this is expressed on the most of the cell on the plasma membrane. Basic feature of this aquaporin structure has been studied in a very detail by the many studies like the mutagenesis, epitope tagging, freeze fracture technique, spectroscopic techniques, and this is how we come to know this is a homotetramer in which every single monomer is consist of 6 membrane spanning alpha helical structures. That means it is going to pass through the membrane 6 times. Such a 4 molecules going to comprise the aquaporins, and both the carboxy and the amino terms of terminus of this protein is going to lie on the cytoplasmic side. If you look at this protein, these are the repeats, you're going to see that. And if you just look at these two domain, NPA domain, what one which is seen on the B, another seen on the E. These 3 amino acids are going to be very, very important. N stands for arginine, P for the proline, and A for the alanine. These 3 proteins are, these 3 amino acids are going to be very important in order to select the water molecule. So they are going to be the part of the selectivity filter which the membrane is going to have. Now this NPA domain, there'll be 2, as I told you, they're playing a very important role in the water selective part of this channel, and orientation of the water molecule moving through the channel assure that the only water passes between the cells due to the formation of the single line of the water molecule. That means the cohesive property of the water, which is due to the hydrogen bond. And you can see there'll be 2 kind of the selectivity filter you're going to see. One is the aromatic, another one is the arginine, which play a very important role so that only the water molecule can enter and the rest of the molecule will not be allowed to come inside. Now, if you see this, the aquaporin, as I told you in the beginning, they have to serve several functions because they're very, very selective for the water. There'll be some other molecule can pass through it, and the protein part, which is having this specific amino acids, which is flanking towards the inner side, will play a very important role in this process. They have many functions to do. For example, they can be present in the kidney, especially the epithelial cell, both on the distal, as DCT we call it as, and the collecting tubule. And whenever the ADH is going to increase, they will allow the more aquaporin to be expressed on these regions so that the water reabsorption can take place. Again, getting of the aquaporin is necessary to keep cells hydrated during the period of drought, and this mechanism involves dephosphorylation on the specific serine residues. That means it's going to be O phosphorylation, and which will bring certain changes on the conformation of the protein. This change in the sharp shape causes the tunnel of the protein to be closed. That means if the phosphorylation is going to happen, they're going to be open, and if the dephosphorylation is going to happen, they're going to be closed. And some of the oocytes where it has been worked in very detail. In oocytes, it transports glycerol and to some extent urea along with the water. That means the 2 more things which can pass through this channel especially in the oocytes. One is a glycerol, another is going to be urea. Again, aquaporins can be used as a target for the drug discovery. Aquaporins mediate water movement into the antral follicle and play important role in the follicular development. Antrum region is the empty spaces which occurs during the follicular development. AQP are known to involved in the early stages of the spermatogenesis as well, and in the secretion of the tubule liquid and in the concentration of the storage of the spermatozoa. Aquaporins take part in the process of the fertilization, blastocyst formation, and their implantation also require the aquaporin to be present. Now, the importance of the aquaporin is going to play a big role in the various aspects of the normal physiology of the body. There are a few aquaporins known so far. For example, 1, 2, 4, 5, and 8. And they're functioning basically as a bidirectional water selective transport. And some of them, as I told you in the beginning, they're also involved in the glycerol transport. Again, whenever the transport through the epithelial cell occur, this is going to be on the basis of active near isomolar fluid transport, such as the fluid absorption by the kidney on the PCT, fluid secretion by the salivary gland on the acinar epithelium. Active transepithelial fluid transport involves the generation of the osmotic gradient by the active solute transport. The high epithelial water permeability conferred by the AQP increase the net fluid transport. Now, aquaporin, the kind of aquaporin that we have in the brain, that is called as aquaporin 4. They are involved in the brain swelling, but it is the astrocytes rather than the epithelial cells that are involved. AQP4, they are also involved in the neural signaling. AQP are expressed in the electrically excitable tissues in the supportive cell adjacent to the excitable cells, and the AQP4 dependent water permeability, which enhances the potassium ion transport by a pseudo solvent drag mechanism. And this neuroexcitation involves ion ion buffering in the extracellular fluid. Other uses of the aquaporins, they play important role in the cell migration. And aquaporins, they facilitate the cell migration, appears to be the general phenomenon relevant in the angiogenesis, that is a part of the cancer, tumor spread, that is a metagenesis, wound healing, and immune cell chemotaxis. There also we have the aquaporins to be used. The two aquaporins which are important in hydrating the skin, one is aquaporin 3, another one is 7. 3 is also involved in the cell proliferation, and 7 is also involved in the fat metabolism. Now, there is some disease which can arise because of the problem in the aquaporins, that is called the Aquaporinopathies. They are, the mutation due to which it will arise. They are, the very rare, though. One of the most common aquaporinopathy is the one caused by the mutation in the aquaporin 2, which cause a non-X-linked nephrogenic diabetes insipidus, we call the NDI. The incidence of the NDI caused by the aquaporin 2 mutation is fewer than 1 in 20 million births. That means it is going to be hugely rare. Another mutation in the aquaporin 0 gene in the lens fiber causes the congenital cataract by a mechanism that is speculated to involve the defective cell-to-cell adhesion rather than impaired water transport. Aquaporin and its correlation to obesity. This is the one topic which has been walked, quite in detail in the recent time. So they do known to be causing certain, the problems with the obesity. The aquaporin selectively conduct water molecules in and out of the cell, while preventing the passage of the ions and other solute, also known as water channels. And some of the aquaporin, I told you already, they are also called as the aquaglyceroporins, which also transport some small uncharged solute, such as the glycerol, sometimes maybe CO2, maybe ammonia, maybe urea. They do play important role in the physiology of the body. A subset of aquaporins, which is known to be aquaglyceroporins, like the aquaporin 3, 7, and 9, they transport both water and glycerol. And the AQP7 express in the plasma membrane of the adipocytes. And usually, if you look at the difference between the aquaglyceroporins, they have less constricted pore size. In the normal circumstances, the pore size is going to be 2.8 angstrom, whereas in this case is going to be 3.4. And they do have some of the hydrophobic regions as well. And aquaporin 9, they are playing important role in the hepatic glycerol uptake as well. For example, if you just work out, if we're going to make a null mice manifest, especially AQP7, that causes the adiposity, hypertrophy, that will reduce the plasma membrane glycerol permeability, cellular glycerol or triglycerol accumulation will take place that could activate the glycerol kinase enzyme for the upregulation. And the certain recent studies which are known, they do play important role in the several physiological activities. The disorder or maybe deficiency or absence of the aquaporin may cause several problems. For example, like the condition I told you before as well, we call as a nephrogenic diabetes insipidus, NDI, which is known to be causing by the aquaporin 2 genes. Then we have the congenital cataract because of the aquaporin 0 genes or O genes. And then we have the one autoimmune disease that is called as Davick's disease, that is aquaporin 4, and which basically attacks the optical nerves and the spinal cord. So these are the few roles that is played by the aquaporins, and aquaporins are one which will increase the osmosis of the water in a large extent, which is very, very important for the many cell to uptake the water in a passive way. So hopefully this lecture will help you. Do share, subscribe, and watch my channel for the new upcoming videos. Thanks a lot for listening me. We'll talk about in the next lecture in the coming few days. Thanks a lot.